the Congress. I sent you the form to vote for the next edition. If you didn't receive it, please tell me, so I will send you again. This morning will be run by two chairmen, Luc Willems from the Naturalis and Josip Skeio from the University of Zagreb. Please.
Yes, good morning, everyone. Josip and I will share this session this morning, and I will kick off, and Josip will share after the coffee break. So I hope everybody is fresh and ready for a new day, full of interesting talks. The first one will be by Nefeli Kotica. I met her some 5 years ago in Crete when I visited the museum, and then she was busy, very busy with examining the site, what do you call it, the pitfall catches from about 40 years across Crete, and she was shifting through all kinds of Orthoptera found in these patches. And so after that, she is done, and meanwhile, she's done her MSc, and now she is busy with her PhD, and she's going to tell you about, her topic is hopping across the mountain tops of the South Balkans. She's going to talk about the phylogeny of the genus Orthoptera.
Hello, can you hear me? Is this okay? Yes. I hope so. Yes. Okay, hello, my name is Nefeli, and today I want to present to you some of our preliminary results on the phylogeny of the genus Orthoptera. As many of you know, the Southern Balkan Peninsula is a biodiversity hotspot with complex geological history and geomorphology that leads to and has many long mountain ranges with many high altitude summits. These are often isolated, forming sky islands and hosting a plethora of endemic species, and thus present great natural laboratories for evolutionary studies. One of the inhabitants of these sky islands, of the sky islands of the Western Balkan Peninsula, is the grasshopper genus Orthoptera, which is restricted to the high altitudes, usually above 1,500 meters, of the Pindos Range, Peloponnese, Albania, and North Macedonia. Orthoptera is an apterous genus with low dispersal abilities, which combine to its requirements for specific mountainous climate and the complex geological history, and complex geological history and geomorphology of the area, results in many isolated populations with restricted ranges. Currently, it consists of 10 described species, plus a number of populations with unclear taxonomic status. Thus, the aim of this study is to define the phylogenetic relationships of Orthoptera populations originating from different mountains based on molecular methods, and to discuss hypotheses about their evolutionary history. Orthoptera specimens were collected from 26 mountains of Peloponnese, Central Greece, the Pindos Range, South Albania, and North Macedonia during 2 field trips in 2021 and 2022. Collected specimens were preserved with ethanol and stored in a freezer. Total DNA was extracted from the high lengths from specimens from each of the mountains using DNA kits. Some Podismini genera were used as outgroups. Using Sanger sequencing, we amplified one mitochondrial genetic marker, NADH2, and the phylogenies were reconstructed using Bayesian inference and maximum likelihood analysis. In order to estimate divergence times in Beast and Beauty, we used the molecular clock based on the evolutionary rate for Apterus orthopterans, calculated by Chang et al. In 2020. Here I should mention, we used a strict model, which means that maybe older times will be biased, are biased. Moving on to the results, first I would like to show you a map with all the mountains that their taxonomic status is unclear right now, and yes. As you can see, they're mostly centered in central and southern Pindos and central Greece. So this is our tree, the Bayesian inference tree. And as you can see, we have two main clades, one Peloponnesian clade and one North clade, which includes the populations of central Greece and Pindos and the Dinaric Alps. Zooming into the Peloponnese first, we have the Oropodisma helmosi clade, which includes the species Oropodisma helmosi, Kilini, and Erymanthosi, whose relationships are not clearly resolved yet. And the lineage of Taigetos, however, the southernmost mountain, is very clearly distinct from the others. Now looking at the North clade, we see two main subclades, which are geographically distinct. One includes all the specimens from central Greece, the northernmost being Mount Timphristos, and the other, all the populations from Pindos and the Dinaric Alps. Yeah, that's it. So, zooming in again in central Greece first, we have two clades, the Oropodisma willemsei and the Oropodisma timphristosi. Here, we notice some discrepancy between morphological observations and mitochondrial DNA history, with more, how do I say it? Like, the most important case being that of this population from Iti, which has been identified as Oropodisma timphristosi, but the mitochondrial tree groups it clearly with Oropodisma willemsei. Moving on to the north part of the northern clade, Pindo-Dinaric clade. Here we have at the north we have populations of the species Oropodisma macedonica, and then in the center is the most interesting part, and unfortunately the one we have the least information of, because we will make some extra visits in those mountains, and also some different molecular techniques will be required to obtain a stronger phylogenetic signal than just that one molecular marker we used. So let's now discuss a bit about divergence times, which were produced by BEAST. I should point out that these are preliminary results, and they will be fine-tuned and developed further soon. First of all, we notice that all the larger clades have quite, which correlate with the distinct geographical entities we mentioned before, like the Peloponnese, have diverged very early from each other during the Pliocene. Interestingly enough, Taigetos, the southernmost Peloponnesian mountain, also has diverged quite very early, about 5.3 million years ago. Another interesting point is that the divergence times between geographically close mountains are mostly around or younger the mid-Brunhes transition of the Pleistocene, about 403,000 years ago. The mid-Bronchius transition is a period which after it, the interglacial periods became warmer, which could have led to the isolation of Oropodisma populations. Some examples of this are the Oropodismas from Tymphristos that separated from the populations from Vardusia and Oxia, as well as the between the mountains that host Oropodisma macedonica. But why would warmer interglacial periods lead to isolation of populations? Well, basically, during a glacial period, an alpine species like Oropodisma would spread to the lowlands and be able to colonize other mountains or gather in refugia. Genetic flow between the populations is induced, leading to hybridizations and speciation reversal processes. However, in interglacial periods that glaciers retreat and the climate is warmer, the alpine species are trapped and isolated on the mountaintops with cold, nice climate for them. And they cannot go to the warmer lowlands. As a result, we have genetic divergence and speciation. These opposing processes throughout the Pleistocene glacial cycles have played an important role in the evolutionary history and current distribution patterns of Oropodisma, as is the case for numerous organisms in the area, including the bush cricket genus Parnassiana, isopods, plants, and others. Other grasshopper genera of Mediterranean sky islands have also been shown to be affected by glacial cycles in similar or different ways, actually. Concluding this presentation, until recently, Oropodisma had been considered only taxonomically, but recently the genus is gaining attention due to its sky island distribution, high conservational value, and its response to climate change. I would like to thank the Ferranchi Fund of the Hellenic Zoological Society for the funding of this project, as well as the Biodiversity Conservation Lab of the University of Ioannina for their constant help and support. And that's it. Thank you for your attention.
Thank you very much. It was a very interesting presentation, especially for someone who knows all these mountaintops. Questions? Any questions for Nefeli?
There's one.
Oh, sorry, Paul.
I have a simple question. If you considered also the genus Ethanopolisma in the general phylogeny, and if there are localities where more than one species of Oropodisma are present.
So far, I haven't detected any such cases in Oropodisma, but in Parnassiana, we had some cases like this, it has similar like sky island distribution.
And about Ethanopolisma?
I haven't worked with it. Yeah.
One short question.
Yes.
Is it possible when you have this women, say, and sister species, is it possible that it's mitochondrial progression, like hybridization?
Yeah, why not? There are two mountains that are very close to each other, so yeah, very likely. I hadn't had time to check to clearly check the morphology yet, because I'm just starting to learn like the taxonomy of grasshoppers, so I still need some time for that.
Do you have any thoughts about why they did not spread further north? Because there is more mountains going to the north?
I don't know, honestly. Maybe we haven't found them yet, but I don't know. Yeah.
Any more questions for Nefeli? No? Thanks very much.
Thank you. Bye.
next speaker is not, as in the program announced, Sylvia Pina, because her presentation is represented by a poster. So we now shift to Axel. Well, I think I don't need to introduce Axel. Axel is world famous in most respects. World famous. And now he's going to talk about a national red list of the Orthoptera of Luxembourg.
So now I can speak 30 minutes? But maybe before I start, there were two things. Filippo made the point that he sent around a poll for the next venue. And I think if there are volunteers here who would like to do it, it would be good if they shout so that we make the right choice in the end. And I think some ideas would be maybe Croatia or Spain or Switzerland, I don't know. So, but maybe you think about it, and maybe after the coffee break you can make a point then. A second thing I wanted to mention is, I mean, I have been chairing the Grasshopper Specialist Group since 2010, and I think it's actually time to step down. And the problem is to find a good successor. And I tried to do this already by electing or selecting a co-chair a couple of years ago. Mark Bushell was the first one, but then he became a co-chair of the Spider and Scorpion Specialist Group. Then I chose Kate Umbers from Australia, and she founded a new NGO, so she's now very busy with her NGO. So if anyone has a good idea or wants to propose him or herself, please contact me, because, well, I think I can make another quadrennium of 4 years, but together with the co-chair, and I would like to hand it over to the other co-chair then. Okay, and you may have noticed that I'm not as busy anymore as a chair as I used to be, because I have so many other obligations. So that was a short introduction. But now we come to my presentation, the National Red List of Orthoptera for Luxembourg. You may be a little bit surprised. I mean, Luxembourg is such a small country. Does it need a national red list, in fact? And I think yes, because red lists are really a very powerful tool to inform conservation action. And I see it in my my home country, Germany, where the information on the red list status of species is always considered when it comes to decisions in conservation. Yeah? So it's really an important information. And if you look at the ISN red list, it provides a lot of information, not only on the conservation status, but also on the distribution, on the ecology, on threats. And that is exactly the information which is needed to inform conservation action. And maybe the large project, which was already mentioned by Armin and others, I think Luc also had this in his presentation, is the European Red List of Orthoptera, which we have completed in 2016, and it's supposed to be reassessed every 10 years. So we assume that there will be a new project within the next 2 or 3 years started to reassess all the European Orthoptera species. And now, I think, since that time, we really have a lot of new information on the distribution. We even have a couple of new species which have been described from Europe, and I think we will have even better distribution maps than we had before. When we started with this red list, the European red list, there were no European distribution maps at all. And we had to get all the information from different countries, and I know that there are some, not so good maps in some of, for some of the species, but they have to improve, and this is a good opportunity to do so. So a quarter of the European species is threatened with extinction, and of course, if you go to a very small scale, like Luxembourg, a much higher proportion is threatened, because if you the smaller the scale you choose, the higher is the probability that species went already extinct. Yeah, and there is actually a red list which was published in 2003, so it's more than 20 years ago. So there's an urgent need for a new red list, and there's also even an atlas for Luxembourg by Roland Preuss and Mark Meyer, published in 2004. So this was actually a good basis for us to start with a new red list, because we thought, okay, maybe we can look for the changes since that time, because Roland Preuss surveyed 200 sites overall over Luxembourg, and we have chosen 100 of those to repeat his study, and Roland Preuss is still involved in this project, so he knows the sites very well. So we have a a time frame of 20 years and see how the species have changed. The surveys will be done or have been done in 2023 and this year, so we already have 56 of the 100 sites more or less completed. We have to revisit some of the sites because the weather conditions were not good or it was already late in the season or so. But this is just a preliminary result. So the average number of species per site in the old times, so in 2000 to 2004, was 9.4. And now we are at 10.3. So actually the diversity increases, which sounds good. Biodiversity increases, it doesn't decrease. And actually we have a very similar study which we made a couple of years ago by my PhD student Sophie Ogan. She studied nearly 200 sites in Rhineland-Palatinate, which is just next to Luxembourg. So let me just check if I can show you. Yeah. So actually the border to Luxembourg is here. Yeah. So it's very good to compare these results. And again, oh, sorry, I went in the wrong direction. The results from this study showed there was even an increase from 8.7 to 11.1. But it's a longer time frame. We're talking about 30 years here, while in Luxembourg we're just talking about 20 years, and the results are not yet completed. So again, we see an increase in species numbers. Everything fine, apparently, but not really, because one thing which these numbers doesn't tell you is the decline in beta diversity. And I made this graph once for a public talk, so for laymen to understand what is meant with a decrease in beta diversity. So when we went in the 80s to these sites, we found in each site different species, represented by different colors here. If we go on the same sites today, we find more species, but we always find the same species. So, and if you now compare the colors above in the 80s with the colors in the in this years, you see that a couple of species just went extinct. So even if we have an increase in alpha diversity, there can be a decrease in beta diversity, even gamma diversity. It's not as bad as represented here, but this is a process we call biotic homogenization, and this is actually found across several biodiversity studies all over the world. Yeah, that we always find the same communities. We find generalists which are expanding everywhere. We find always the same species communities, which is driven by the same procedures of land use all over Europe, the same fertilizers, same pesticides, same machines, same livestock, and of course invasive species as well. Another interesting result, sorry, it's difficult to find the right point here, was that we also found some altitudinal range shifts, which is an indication of the warming climate, of course. And this one here is Homocestus viridulus. Again, this is the data from Rhineland-Palatinate. And you can see the gray bars is the altitudinal distribution from 1986 to 1999. Most of the data from 1989, 1990 around. And the black bars are the ones from the current situation. And you can see actually a strong decrease in the lower altitudes, but in the higher altitudes it remains similar. So the arrow at the top shows you the shift of the mean altitude. So we see an average increase in altitudinal distribution of the species, which is in this case only caused by the decline in the lower altitudes. And this decline was about 28%, which is quite strong. And if we now look at Luxembourg, we have of course, not as a strong altitudinal range here. So actually the highest mountain if we want to call it mountain, in Luxembourg is 560 meters, which is more or less this red bar here. And you can see there's not really these high altitudinal distributions where the populations still remain constant, well, they hardly exist there. And as a consequence, the decline of this species in Luxembourg was 59%. We have another species which shifted in altitude, which is Oediputa caerulescens. You can also see the arrow at the top, which means there is an increase in the mean altitude. But you see there's a completely different pattern here. Yeah, the black bars are always higher than the gray bars, so it's expanding everywhere. In the low altitudes, in the high altitudes, and the difference is in former times, the few localities at higher altitudes were in very special habitats, like quarries, which have a very warm microclimate. And now we see they can expand from there to less microclimatic warm habitats like railroad edges or something like that. Yeah, and so they are not as specialized anymore. This is a pattern we see in a lot of species. Oediputta carolatens was on the red list in this part of Germany and also in Luxembourg in the past. It's not anymore. And we see a lot of other species which expand in a similar way. So this has increased by 330% in this part of Germany. And I said these were about 33 years actually. In Luxembourg, we just talk about 20 years. And in fact, we have 20% increase, which means that we have more or less 10% increase per decade. Now, if we look at some special species here, the warbiter bush cricket is one which is, of course, threatened in large parts of Central and Northern Europe. And you can see this is a historic distribution of the warbiter in Luxembourg. And the current distribution since 2010 looks like this. So we have only a very few localities in this part of Luxembourg where we have still some chalk grasslands left. The species which probably declined most was Omocestus rufipes in both studies, and I was surprised to hear that the same was true in Austria. And it's a little bit surprising because this is a species which is so common in the Mediterranean. So we would expect it to expand, so why doesn't it expand? And I have the impression in the Mediterranean, if you go to Italy or somewhere else, you find two generations of this species. You find one in the spring and one in autumn. In Central Europe, it has only one in summer. So when we have very warm years, it might happen that the first generation comes very early and it tries, or maybe a second generation just starts and then the winter comes. It's just a hypothesis, but this could maybe explain why this species goes extinct or declines in northern latitudes. So also 59% decrease. And then we see some species which are also expanding. This was a species which is completely new for Luxembourg, Calyptomus italicus. A species which was also in the neighboring countries red listed. In Germany, it was critically endangered. There were only very few populations left a couple of decades ago. And now it has expanded massively. And it's even found in mid-altitudes, montane and colline situations, so very surprising. This is the current distribution in Luxembourg. You can see most of the records are here from the southwest, which is the warmest part, the Minette, with a lot of old quarries and mining areas. And then you even find them here, and this point is particularly interesting because it's already in the colder part, in the Luxembourgish Ardennes, which is called the Ösling. And in fact, we find similar records here in Germany, in the Eifel Mountains, which is also quite surprising. Another species which has increased dramatically is Cortipus dorsatus. That is a species which was in the neighboring Rhineland-Palatinate. We had only 5 populations in the 80s. Yeah, and now we had it on 80% of the sites. And a similar pattern is here in Luxembourg. As I told you, we have studied so far 56 sites, and it's present on 39 sites now. Also a species which is a little bit late in the year, so maybe it's benefiting from the longer vegetation period, and so that it simply can complete its development within the season. One species which is also surprising that it's declining is Bicolorana bicolor, because this is a species which appeared just in the 90s more or less, and has spread in large parts of Germany in the 90s. So everybody thought this is also one of the winners of climate change, but in fact now it's decreasing. We are not completely clear why. One hypothesis is that it has problem with the fertilization of the vegetation because it needs some bare ground to oviposit, so it's some heterogeneous vegetation, and because of all these fertilizers which come from the air, the vegetation is now more dense, but that's just an idea. Another decreasing species, Pseudocarpetipus montanus, of course a wetland species, and this one is the one which needs permanent wet soils, and because of the strong droughts we had in the last years, it's declining. We had a good study on this by a former PhD student of mine, and the same pattern is seen here in Luxembourg. Actually, one threat we sometimes forget is land abandonment. We often talk about agriculture, and we already heard that yesterday that in the Alps, land abandonment is also a major threat because as soon as we lose the small farming systems in these areas, they are overgrown by shrubs and forbs, and in this case, we cannot find, the right one was a former habitat of Pseudocalyptus montanus, which is just lost because of this strong growth of vegetation. And it's the small farmers which were managing these sites before, yeah, and we lose small farmers, so the loss of farmers is going hand in hand with biodiversity loss. So let me come to the conclusions first. The red lists are, of course, very important because they provide information on the conservation status of species, and they are even important for small countries like Luxembourg because they identify the major threats and can, of course, guide conservation action. The extinction processes are, of course, much faster at a smaller spatial scale, and therefore there's even a more urgent need for starting conservation action. And we will complete the final field work in this season, maybe in the next, because this season was very rainy this year so far. And then we aim to publish the red list in 2025 or 2026 and hope that it will stimulate conservation action. So thank you for your attention.
Thank you, Axel. A small country, a lot of results. Questions from the audience. Amin.
Thank you, Axel. Overall, I think I see the same inconsistent results, which doesn't only relate to climate warming. I mean, you have also very thermophilic species decreasing and increasing, of course, and mesophilic species which are decreasing and which are increasing. You did mention just for one sentence the artificial nidification of habitats. The microclimate cooling of such habitats might be much more important than any temperature rise, which is on a global scale. For instance, Orthoptera ruficus, to my experience, is heavily impaired by this microclimate cooling in these landscapes. And also, I cannot explain why Dactylus versivorus should be somehow impaired by climate warming. It's a very thermophilic species. And another thing I would like to stress is, I mean, I'm a bit amused, of course, to hear where altitudinal redshifts in a country with only 300 meters of altitudinal differences. One should really show that at this site, not at a global scale or whatever, the temperatures between, say, 300 meters sea level and 500 meters sea level have really relevantly changed before one speaks of influences of climate warming. And you should more, maybe, I think Luxembourg has good data about the landscape changes, about the agricultural changes. You talked in the last part of the talk about the abandonment of habitats. Of course, it's a working process, but I'm a bit tired of this constant climate warming hysteria.
Well, well, I don't say it's hysteria, because we will have more species because of climate warming, that's quite clear. First of all, it's not the only threat, but it's becoming more and more prominent. I told you about the land abandonment, which is a major threat for Dactylus, for example. Dactylus has, in my opinion, not really a large problem with, but, well, if you look at the southern distribution of Dactylus, you find it mainly in montane and alpine areas, and not so in the lower lowlands. And we have some low-living populations. We will see how they will develop in the next decades. But currently, I don't see a pattern here. And Orthoptera ruficus, it's a different story, because these sites where it went extinct look still perfectly fine, with a lot of bare ground, yeah, which is the structure it needs. And that's surprising, yeah. While it actually survived in places which looked to me, not ideal anymore, which were too dense in vegetation. So, which doesn't really fit to the, the microclimatic preferences or the former microclimatic preferences. And we see a lot of species which are now expanding and shifting in the habitat, like I told for Oedipoda cavalescens. The altitudinal range shifts I showed here were not from Luxembourg, but from Rhineland-Palatinate, which has only 900 meters, not so high, but still, it's interesting to see. These were just those which were significant, and you could see this in the graph, the very strong decline in lower altitudes, which we not only had for Homocestus viridulus, but also for a couple of other species, Metrioptera parhyptera, Myrmeletetix maculatus. So a lot of these more cold-adapted species, Homocestus viridulus is also one of them. Yeah, and well, I wouldn't say that climate change will lead to a decline, an overall decline in species diversity, because new species are coming already because of that. But it's a factor which is definitely plays a more and more prominent role. Yeah. Okay. Sorry.
Very short question, Thomas. You had a question or not?
I just wanted to say that it was surprising for me how similar the situation in Luxembourg is with Austria.
Yeah.
We did also many such revisits on sites, and I would have selected the same species that you selected to demonstrate what happens in Austria. It's really interesting. Also,
yeah, it's actually. And Homocestus viridulus is also declining in Britain. Yeah, so it appears to decline all over Europe.
Okay.
The factor with the nitrification is, I think, also a very important thing. We could prove that the group, we said approximately 20% of the Austrian species belong to very nutrient-poor habitats, and this is the group with the sharpest decrease.
Yeah.
Okay.
So
Yeah. It's a really rare response. Yeah.
might be also for, I agree with the
Okay.
assessment of Victor Varane.
Okay. Okay. Thanks. Thank you, Axel. Okay. Our next presenter is Thomas Zunakratki. He's from a lecturer in Vienna, or not? Freelance, okay. Landscape ecologist, sorry. Of course, he's also the author of the, in 2017, of the Grasshoppers of Austria, so he's very knowledgeable about the Grasshoppers of Austria. And now, today, he's going to present some new insights on the distribution of Ausentreit's bow-winged grasshopper, Chotippus eisentrauti, in the Eastern Alps. Please, Thomas.
Yes, thank you very much. I'm happy to present the results of very intensive field work by Inge Illich. She's here in the auditorium. And the results of the analysis she did together with Brigitte Gottsberger, an expert in bioacoustic, especially of the genus Chotippus, and me, who is mainly just the talking here now. I hope it's, so where do I, yes. The species, it's quite an interesting species. We are in the group Chotippus. Everyone knows it's a very complicated group. It's a young group with lots of cryptic species. Chotippus eisentrauti, the Eisentraut's bow-winged grasshopper, was already described by Willy Ramme in the early 1930s from Austria, from the southern part of Austria, from Carinthia. Species found in very interesting place and was believed to be a good species. But there was quite a lot of debate, especially in the 90s. Is it really a good species? Isn't it just some ecomorph of Bigutulus? But fortunately, in the last years, there were also work done, for instance, by Oliver Havlicek, who could prove that Eisentrauti is definitely a good species. You see, yeah, a typical Chotippus, quite prominent in coloration. And the German name is Südalpen Grashüpfer, so the the grasshopper of the southern Alps, because it was always believed that it replaces Cordypus bigutulus on the southern edge of the Alps. And the strange thing, in the last 10, 15 years, we had several observations of strange bigutulus in the northern part. You see here Austria, and the red dots are the recent observations from Eisentraut in the southern part, south of the Alpine main ridge. And these white spots in the northern Alps, they were quite strange. So for instance, we got this photograph of an Cordypus bigutulus, but it looked like Eisentraut and came from, definitely not from south of the Alps. So there was much debate, there was much confusion, and we wanted to know what's really going on. And Inge, who is the main driver behind this question, she visited 27 mountain ridges in Austria in the last 10 to 15 years, mainly in the last years, to collect data on this Cordypus eisentrauti, this dubious eisentrauti, but also syntopic and sympatric specimens of Bigutulus and Bruneus. And she collected specimens and she recorded songs. We had 3 hypotheses. The northern ones, you see the yellow dots, all the yellow dots were visited by Inge in the last years. In the northern part of the Alps are only ecomorphs of Bigutulus who look quite bright, and so the other hypothesis is no, there are relict populations of Eisentrauti from former larger distribution. And the last one is it's just some hybrid from earlier times now integrating, and you can't distinguish them between Bigutulus and Eisentrauti. You see the yellow points are from north of the Alpine main ridge, and the red ones are samples from south of the Alpine main ridge. The habitats are quite dangerous, so you can follow Inge into the habitats of Eisentrauti. I'll show you some of them. This one, it can be like this. It can be like this. Beautiful habitats. And it can be like this, you know, one hop and you make a really big jump. And also the habitats in, in Italy look quite similar. This is from Tolmezzo, also here. Inge found Cordypus eisentrauti, south of the Alps. We did measurements and analyzed and compared it with between the population south and north of the Alpine ridge. We compared it with Bigutulus in the same areas and with Bruneus. The most important morphological character was the ratio between the coastal and the subcoastal field. You see there is no significant difference between the northern and the southern population, but highly significant difference between Bigutulus and Bruneus. The other very good morphological character was the width of the head. So Eisentrauti has a bull head compared to Bigutulus, and you see also no difference between north and south, but very significant difference between Bigutulus and these both populations. We also looked at the wing length, and here we see, that's interesting, we have significant differences between all 4 groups, also between north and south of the Alpine ridge. So there seems to be also some difference. Bruneus definitely has the longest wings, but Eisentrauti is also a bit longer winged than Bigutulus. How about the acoustic differentiation? The main character is the length of the verse. It's much shorter in Eisentrauti than in Bigutulus. This is very good field feature. If you know the song of Bigutulus very well, you will rapidly recognize there is something different in Eisentrauti. And what's also typical for Eisentraut, it has a higher number of verses, up to 13 in a song, and Bigutulus is much, it's quicker with its song. It's finished after, on average, after 3 verses. So you see, don't know if can I show something? No. You see the quick verses, the short verses of the both groups of the Eisentrautis, and the longer and only few verses by Bigutulus. I hope we can hear the song. I added one recording. Let's see. So this was Eisentrauti, short verses, and we heard it 4 times. So we conclude that we have the same species, the same taxon, north and south of the main alpine ridge. They look quite similar, they have similar measurements, they don't differ from each other, but they differ significantly from Bigutulus, acoustically and morphologically. There is another important difference. You know, when I showed you the habitats, Cortipus eisentrauti lives in habitats that are very steep, very sun exposed. They have an extreme microclimate, and this it's too hot to dry for Bigutulus. Bigutulus it's a very aeroic species. But even in Austria, in extreme habitats like in the Lake Neusiedl area or in the sand dunes of the Machfeld, Cortipus bigutulus is missing due to microclimatic circumstances, and the same seems to be true for these extreme habitats in the Alps, where it's the main co-syntopic species were Gomphoceribus rufus, Pholidoptera, Aptera, and Cortipus bruneus. There's a very interesting poster by Robin and colleagues in the poster session where where also specimens from north and south of the Austrian Alps and from northern Italy and from Berchtesgaden in Germany were assessed with phylogenomic analysis, and it proved the situation we found here. The thing is, how could this species, this Eisentrauti, who is definitely not that aeroic as a Bigutulus, survive in this large soup of Bigutulus? Bigutulus is everywhere in Central Europe, and you have these islands of Eisentrauti. Hybridization is possible. It's also proven, you can see it in the poster, it was proven interestingly in the area of the locus typicus. So it would disappear inside of Eisentrauti, but we think the main reason is this very specialized habitat choice in combination with a different acoustic communication. But these steep sun-exposed limestone cliffs, these scree fields, they are very, very extreme. We looked also at the studied mountain plots. Where are the occurrences in elevation? And Eisentrauti is significantly higher up in the mountains than Bigutulus on the same mountain sites. And these sites are also very rare in other Orthoptera species. On average, only 2 to 3 different additional species can be found there. So it's an area where the competition is very low. So a picture like this is an exception when you find Eisentrauti together on a very attractive feeding place with Gomphocerus sibiricus, Pseudocortipus parallelus, and Metrioptera saussuriana on one of the places in Salzburg. And just the end, we wanted to think about how did this quite strange distribution pattern appear. We compared it with other distribution patterns we found on animals and plants in the Eastern Alps. It's quite unique, this northern and southern distribution with this big gap in between of some 50, 60 kilometers at minimum. We think that glacial refuges, like we heard it yesterday from Anonkonotus, play a major role. I depict here in yellow supposed botanical refuges by Schönswetter, who proposed also a refuge in the northeastern part of the Alps, which might be the refuge for the northern populations, and the Italian pre-alpine refuges, which most probably will be the refuges for the southern populations. Yeah, thank you for your attention.
Thanks very much, Thomas.
Welcome.
Any questions? Joseph?
Could you please put the map?
Yeah, this is interesting point.
It's interesting because
A moment, which one is it?
morphological and strong Eisentraut in Istria. But
Yeah.
what we have in Rovereto in very similar habitats where I study Ramihipus is something that we call Orthoptera bigutulus durmitor form. They
Yeah.
from Montenegro. And I think it's either sister species to Eisentrauti or it's Eisentrauti widespread in the Dinaric Alps.
Mm-hmm.
It would be nice, we recognize it from bigutulus by this shorter version, old dirty song.
Yeah.
Bigutulus.
Okay.
For now, we included Eisentraut in the checklist, but it's very interesting what will be this dynamic.
Yeah. I think this the Bigutulus group is still full with surprises. So when I think there is this Hedicki and the Euhedicki and this Domitov, these are all cryptic species in Bigutulus, which I think they all have in common that they have a bit different song and a strikingly different habitat, maybe. Yes. So they all have the problem how they have this super Bigutulus. He can be everywhere, and he's still so close that they can hybridize. So what's their strategy to survive this evolutionary pressure?
We have not a lot of time. One question. Yes, we are running out of time. It's your choice. It's your choice. One question.
Yeah, well, I can discuss this later on. This box plot graphs always have a problem. I mean, we just focus on the mean levels and the percentiles, but if you have a closer look, you have always many outliers.
Sure.
What would you make up of this? Are these hints of more hybridization going on?
No, the overlap is dramatic in some features. So you can't, it's often a big problem if you have only one specimen dry or so, it's not possible to say, is it now Eisentraut or Bigutulus? So the best way to identify the species is really in the field, when you see the habitat, when you hear the song, when you see the live animal, and you see several. I think the overlap is because they are still so close, so it's really a very young Bigutulus offspring, and but are stable due to these special features we said already. So they're so close, so it's still morphologically not very sharply differentiated. But the whole, if you take all all samples together, it's a significant difference already.
Thank you very much. I think we are running out of time a little bit, so we have to go to the next. Thank you very much, Thomas. So now our next presenter, before the coffee break, we're running out a little bit of time, but I think it will be okay in the end. Next speaker will be Sebastian Koenig from Munich. And he's going to talk about traits shape the climatic niches of Orthoptera along micro and macro climatic gradients. Case study from Berchtesgaden Alps. The Alps are quite interesting. Okay, please.
Thank you, Luc. So, good morning. I'm happy to meet so many grasshopper enthusiasts here. It's really nice. I love this kind of congresses and talking about grasshoppers a lot. Today I'd like to tell you some results from my work in Berchtesgaden, so in the very south of Germany. It's an alpine region. There are quite pristine landscapes still conserved, so I think it's an area which is void of really intensive agriculture and void of fertilizers in huge amounts, so a natural place where we can still learn a lot about nature and natural dynamics. So, climatic conditions are recognized as very important determinants of species distributions and therefore also for biodiversity patterns. So it's not really surprising that we see this spread of thermophilic species, especially in Central Europe where there's good data. Some species such as the Italian locust or the Italian tree cricket, which are spreading a lot in Germany now. However, we already mentioned that there are some thermophilic species which also decline, even though the macroclimatic temperatures are increasing over time. And one point which I want to make is that it's not only the macroclimatic temperatures which are changing, but microclimate can also play a critical role here. Microclimate is often ignored in species distribution modeling, and this can lead to overestimations of range shifts of species. A good place to study these effects of the microclimate are mountain regions. If you take a look at this picture, you can already see there's so much variation, topographic variation, and also variation in the vegetation, for example. So you can expect a high degree of variation of macroclimatic conditions along the elevational gradient, but also of microclimatic conditions. The same holds true for this picture. It's a thermal image where you can see along the slope of a mountain, you find these very warm regions, even in higher elevations than this forested region down. But also there are different factors which influence these climatic conditions. It's not only the exposure of the slopes or the slope itself, but also the vegetation structure or the shading from different mountains. It's different habitats. This is already data from Berchtesgaden, which I want to show. We measured the microclimate at a lot of sites there. Different habitat types differ in their offset of the microclimatic temperature to the macroclimatic temperature. So this is the deviation of the climatic conditions which the species experience close to the ground. As you can imagine here, this is a steep slope where also Cottypus eisentraut is occurring, reference to the talk before. There we have a high diversity of microclimatic conditions because we have these sun-exposed slopes which get very warm at the surface, but we also find a lot of trees, and in the shade of the trees there can also be some species which, there can also survive some of the species which are not so thermophilic. So following this metabolic theory in ecology, we expected that warmer temperatures will lead to a higher diversity and abundance of Orthoptera, especially because most of the species are quite thermophilic in Central Europe. I want to show you some examples of how our study sites looked. We selected the study sites along an elevational gradient ranging from 600 meters until 2,300 meters. There are very different habitat types like this, lakes and extensive pastures in the valleys. And then further up, we can find these even more extensively used pastures with very low densities of cattle grazing there. In the higher altitudes, a lot of similar sites formerly grazed. A lot of them are abundant now. But we also have these naturally open habitat types which are caused or kept open by natural dynamics in these mountain regions, like mudslides or avalanches. And then we have these high elevation habitats, which are either south exposed or north exposed, so there can also be differences. Good. I'm sorry, now the presentation skipped a bit. We're close.
Please.
Yeah, no. I hope you closed your eyes for a second. So what we did is in-field measurements of microclimatic conditions, and we made use of our dense network of macroclimate stations. And additionally, we also used remote sensing techniques like LiDAR to scan the entire national park. So we could create first maps of light conditions and also via mechanistic modeling of microclimatic conditions across the entire park in a very high resolution. Of course, this is just a modeling approach and not really close to the actual conditions. However, there was already a couple of days ago a paper stating that this including or approximating the microclimatic conditions in a habitat really improves predictions of species distribution models a lot. What we then did is survey the Orthoptera communities, of course, with traditional methods, transect walks, isolation quadrats, and also measured some of the traits of the Orthoptera along the elevation gradient. This is what we found. Quite a lot of species for such a cold region. Almost 40 species of Orthoptera occurring in this region. The most abundant ones were, of course, the common bowwing grasshopper and the meadow grasshopper, but also the common green grasshopper. And some quite rare species and specialists for alpine environments as well. We then used hierarchical joint species distribution modeling to estimate the climatic niches of these insects along the elevational gradient. So on the one hand, the macroclimatic niche, and on the other hand, the microclimatic niche. And we found some differences in these climatic niches from the species. Starting in the top left corner with the field cricket, it was only found in the valleys and only there at sites with a warm microclimate. And then we have another response type like the one from Cautipus eisentrauti, which occurred basically along the entire elevational gradient, so broad macroclimatic niche, however, only at very warm microclimatic sites. However, some of the species, such as the dark bush cricket in the bottom left corner, they really avoided the warm sites, the warm microclimate sites, but only in the valleys. So you can see here already kind of an indication for an interaction between microclimate and macroclimate for the species. Another response type was the one of the green mountain grasshopper, which really never occurred in the valleys. So most of the species had their abundance peaks at low elevations and at sites with a warm microclimate. However, there were also some exceptions like this bog bush cricket, which had no real response to microclimate in our data. These patterns are also reflected at the community level, where we found a decrease in diversity and abundance of grasshoppers or Orthoptera in general with elevation and also with cold microclimate. The community composition was also structured along the elevational gradient, with highest dissimilarity found at intermediate elevations and enhanced by microclimatic contrasts between the study sites. As I told you already, we looked at some traits, and I just want to show you one result which was quite striking, and it was the hatching phenology of the species. The hatching phenology decreased with elevation and cold microclimate. That means that probably only these species which hatch early, they can make use of climatic conditions which are quite cold, so the season is quite short for these species, and this also fits well to the results of the species which hatch later in the year, profiting from recent developments of the microclimate. We even found some intraspecific differences. For example, some of the grasshoppers, they grew smaller at the summits compared to the low elevations. Also probably because the season is shorter up there. As I said already, we don't, one important factor is looking at the microclimate, but now I just showed you results of the average microclimate at the study sites. That's only half of the story because microclimate is really different in very small spatial resolutions. So we also did this modeling approach where we checked the spatial differences in the microclimate and calculated some sort of microclimatic heterogeneity of the study sites. And you can see here that this is also a very important predictor for species richness, meaning that at some of the study sites you can find cold conditions close to very warm conditions, also increasing the species richness at the sites. It then makes sense that these species like the green mountain grasshopper and the common mountain grasshopper can occupy the same study sites. Yeah. Now I was restricting myself to temperature. However, I also wanted to take a look into the moisture conditions at the study site because soil moisture is also a very important determinant for many species. We also had moisture sensors, but the data from the moisture sensors are not really trustful in my opinion. So there needs to be some work. It really depends on how close the soil is to the sensor. If there's a little bit of a gap between them, it really differs. It really makes an impact on the data. So we can't really use them now. So I approached this by calculating the moisture indicators by Ellenberg from the plant community to get an impression of the moisture conditions at the study sites. And what we found here is also at study sites where we found plants which prefer moist conditions and plants which prefer very dry conditions. This has an impact on the species richness of Orthoptera as well. So it's not only the microclimate, but also the moisture heterogeneity at the study sites, which plays an important role in these mountain systems. To sum this community analysis up, we found support that temperature plays a crucial role for Orthoptera, for defining Orthoptera communities. However, our results were also nuanced, as for a fraction of the species we found interactive effects of microclimate and macroclimate, indicating narrower climatic niches of these species than the entire elevational distribution would suggest. That means it's very important, and I pledge to really involve or to try to involve microclimate data in predicting species distributions. Also because this sort of data via modeling approaches is increasingly available for larger parts of the world. However, we could probably expect some sort of increase in species richness, especially in regions like Berchtesgaden, where there is a thermal limitation in the Orthoptera community. Some of the traits of insects, such as the hatching phenology, can make support for helping to predict these responses of species to climate warming. As I said already, increases could be possible. Not all of the species will profit from warming. We found evidence for some of the mountain species already retracting. However, as we have these mountains with a lot of different microclimatic conditions close by and different habitat types, like the forests, we expect that there are still refugia also for the mountain species in the close future. And with that, I'd like to thank you. This is the Cotypus eisentrauti found in Berchtesgaden. So a very beautiful species. I was really happy to meet it there. Good, thank you.
Thank you very much, Sebastian. One question before we go for a coffee break. Who has got a question?
Thank you for this very interesting approach. I think it's very necessary to do so. Anyway, I'm a bit confused because maybe of the number of graphs. Got that right? You showed only the model graphs.
Yeah. exactly, yeah.
Progesterone.
Yeah.
Wouldn't it be wise just in the beginning to show if there are some correlations for some specific species between the microclimate at the site and the grasshopper community or whatever you like to name this? Because I'm sure you know the studies of Christian Kerner, one of the most renowned urban ecologists in Egypt. And then mountain slope there, thermologus like every few meters, and it turned out that for a beetle or a grasshopper, only 4 or 5 meters would mean 40 degrees average temperature difference.
Yeah.
So
yeah. We had two approaches. One was the modeling approach. One was an approach with a lot of study sites and a lot of data lockers. And one approach was putting a lot of data lockers at one site to check the microclimatic variability. And all of these features correlate very good with Orthoptera distribution data and abundance data. However, you're right, I only showed the modeling results here in this study, but I can also prepare some of the other graphs as well. Yeah. Thanks.
Thank you very much. Filippo, I think we have to be back from coffee 11:30. Is that, we stick to the program time? Yeah, okay. So, coffee break, 11:30, we are back here, please.
Are we ready for the next session? So first we are going to hear from Apostolis Stefanidis from Greece University of Ioannina, Distribution Pattern and Conservation Ecology of two globally threatened Orthoptera in Greece. So Stefanidis, podium is yours.
Hello, thank you for having me. I'm going to present you the distribution patterns and conservation ecology of two globally threatened mountain species. The project is funded by the Hellenic Foundation for Research and Innovation and is part of my PhD. Well, some things about Greece first. Well, Greece hosts almost 400 species, and 1/3 of them is endemic, and 37% is threatened. These maps that you already know very well from European Red List of grasshoppers, the more intense the color, the darker the color, the more intense the effect. So as you can see, Greece, as almost all the Mediterranean countries, has high endemism, high percentage of threatened species, and high species richness. Therefore, we targeted two endemic and endangered species of Greece, Parnassiana parnassica and Oropodisma parnassica. Nefeli Kotitsa in the morning told you already some stuff about Oropodisma. Parnassiana lives in the same areas. Generally, those two genera coexist in most of their distribution. And we find these two species in two mountains of Greece, Parnassos, from where the names are derived, and Elikonas. And in the last IUCN assessment back in 2015, Parnassiana was assessed as critically endangered and Oropodisma as endangered. So we set, not all the objectives seems, but we're going to see them one by one. We set 5 objectives so we can learn some more things about the ecology of these species, because as we said yesterday, there are a lot of information, taxonomic information mostly, but not so much about the ecology of threatened species. So we try to achieve these species by visiting the distribution ranges of the species. And each time we found one of the target species, we delineated a quadra, 10 by 10 meters around it. And within the quadra, we recorded microhabitat parameters like grass height and the coverage of the ground, like soil cover, rock cover, stone cover, grass cover. And also we recorded the dominant plant species and the population densities of the target species. So the first objective was to find what is the current species distribution. In the map, you see that the orange is the distribution range of Parnassiana parnassica, and the blue one of Oropodisma parnassica, according to the assessment of IUCN back in 2015. We visited 103 sites within these distributions. We found Parnassiana parnassica in 33 sites and Oropodisma in 36, and their distributions overlapped in 23 sites, almost in 50%. And as you can see, the crosses are where the species were absent, so we found them mostly up in the mountain above 1,500 meters, and in Parnassos we reached at 2,300 meters. The first target was to model the potential distribution of the species using SDMs, and then to quantify the connectivity of the suitable habitat. So for this target, we didn't use the factors that we collected during the quadra sampling, but we downloaded bioclimatic variables from WorldClim, 19 of them, 4 topographic and 1 vegetation related, NDVI. And then we set the same resolution to all of them, 100 by 100 meter cells. So we have a larger resolution for SDMs and finer at 10 by 10 for quadras. And we did an assembly of small models with a random forest algorithm. So these are the maps. Upper is the Parnassos mountain, and downwards is Elikonas. As you can see, the more red the color, the more habitat, the more suitable the habitats. So in the more suitable habitats are in the high altitudes, above 1,800 meters. And in Heliconas, we didn't find many individuals, only 3 sites. So as you can see, there are only a few suitable habitats in a specific summit called Paliovouna. About the fragmentation of these suitable patches, the model showed that Parnassiana parnassica has less suitable patches than Orthoptera parnassica, but they are bigger in area, and also the patch cohesion index was good for both of them. So we conclude that the distributions of the species within the mountains are not severely fragmented, as it was assessed in 2015. The next objective was to investigate the environmental factors that shape suitable habitat, and then the ones from the quadra to see how they regulate the species abundances. So about SDMs, for Parnassiana parnassica, we found that topographic position index, slope, and temperature annual rates were the most significant. The species prefers slightly elevated areas, probably for the thermoregulation, medium slopes, and very small rates of temperature annual range. Orthoptera parnassica had as significant factors NDVI, slope, and potential evapotranspiration. NDVI probably interacts with other variables because it had a flat response curve. It prefers all the same slopes, medium slopes. And we found that it wants 200 millimeters of evapotranspiration, which is the water stress. And the whole study area has between 60 and 240. So the species is in the upper level, and this could be, I don't know, it could be a problem with climate change, and it needs more investigation. Now, to see which factors influence the species population densities, we ran some GLM using the variables that we recorded inside the plot. And here you can see that for each species, the most significant variables were one variable for Parnassiana parnassica was mean grass height, which had a negative impact. Hence, when the grass height is decreasing, the population of Parnassiana parnassica is increasing. And it prefers a grass height of 20 centimeters, which we think that the sheep-goat grazing in the mountain of Parnassos maintains that grass height, and it's good that that doesn't stop, and we won't have any forest encroachment. And the Orthoptera parnassica was infected positively by altitude, and we also see that on the field, where the higher we got, the more abundant were the species. The models explained 20% of the variables. It's a little bit low, but for such a simple model with only one variable, it's adequate. Then we wanted to update the IUCN Red List status. So first, we assessed the threats. Well, this is on Mountain Heliconas. These pictures are from last year. The mountain has wind turbines all over it, and there weren't wind turbines in the upper summit where the species reside. But last year, we saw a new road constructed there and some vehicles, and these are within the habitat. So we already have some habitat loss for these small habitats there, because in Heliconas, as you remember, they have only 1-2 sites, and they're already losing them. And of course, in Greece, unfortunately, each year we have some mega fires, extreme events that happens, and we lose sometimes whole forests or even mountains, and this could happen either on Parnassos or Heliconas and lose the whole populations there. So we use these threats to upgrade the IUCN Red List status of the species. So as we said, these species have a restricted geographic range. The threats are land take, wildfires, and probably global warming, but that needs more investigation. We found them only 2 locations, those 2 mountains, and the 2 mountains cannot communicate, the populations do not communicate. There is not genetic flow between the mountains, only within. And there is a, we inferred a continued decline in the quality of the species habitat. Therefore, the Parnassiana parnassica was assessed as critically endangered. The previous assessors didn't have so much data as we have now, and our analysis showed that the species is not severely fragmented. Therefore, we consider that it should be downgraded to endangered, and Orthoptera parnassica should remain as endangered. Finally, some conservation implications that you cannot see. Okay, here is the mountain of Parnassos. The blue one is the Natura. You see the occurrences of the species, and with the yellow is the roadless area of Parnassos. As roadless areas, we define the areas that have at least a distance of 1 kilometer from a road. In Greece, already the government has banned road construction in 6 Natura 2000 roadless areas. Therefore, we suggest that the Parnassos roadless area should also be included in the zone of absolute nature protection. It is not yet there. Also, the Environmental Agency of Greece should prohibit the extension of artificial surfaces within the suitable habitats. And we also suggest that there should be constant monitoring, even from the management body that is responsible for these areas, so that we can have data about the population trend to know if the populations are increasing or decreasing. And finally, because as we said, there are high elevation species and they also are affected by some climatic variables, we think that it's important to do some SDMs for the future to see how the species are going to respond in the climate change, and that is one of our targets, the next target we have. I'll leave here the main points of my presentation for discussion and questions, and thank you very much.
Thank you very much for the nice lecture. We have time for 2 questions because in this session there will be one lecture less than in the previous.
Hello.
Have you implemented some conservation measures in practice that the people do that or not?
Sorry?
Have you implemented some conservation measures also in the practice?
Not yet.
Or you plan to do that?
We have already informed the Environmental Agency of Greece. We have given them the data, so they know where the species are, and we give them the spots where the species are more abundant, so they know that they should protect these areas from new constructions or expansions of other constructions. But we also want to start do some constant monitoring, collaborating with the management body there. Yes.
I have a slight methodological question about your species distribution model.
Yes.
Which program do you use?
Biomod. Biomod from yes.
Okay, because I was just wondering that you used 3 points for the modeling.
No, 3 points. No. It was 33 for Parnassiana, Parnassica, and 36 for Orthoptera.
Perfect, thanks. And for the variables, did you use it from WordClip or 19, or did you do some variable correlation?
We downloaded them, and it was, I think, from 2000. And then we... Yes. And then we did a variable selection with VIF and Spearman rank correlation, and we keep only 9 for each dataset, for each species.
Okay, because there are 2 layers in WordClip which have a lot of bias and they should be never used. No, 4. 8, 9, 18, and 19. So, yeah, I was
Probably.
just wondering if you used the variables, or I was just interested in the variable correlation.
We did some correlation tests before we used the variables. Yes. Thank you.
Thank you very much. Now, we take 15 minutes break from Orthoptera and we go to Mantodea. So Zohre Mirzae from Persia is our next speaker. She's from Persia, but also she's representing Senckenberg Institute from Münchenberg. The podium is yours.
Thank you. So hello everyone. I'm going to talk about Mantodea. So I hope that you get some interest in Mantodea as well. And so a bit about me. So I am Zohre, I am from Iran, and I've been working on Mantodia of Iran and adjacent areas mostly. And I am mostly interested in biogeography, phylogeography, and systematic. So in this talk, I'm going to talk about Holoptilon, one of the very amazing genus in Mantodia. And this genus contain like species that they are really tiny and small specimens. They are really like, they can be like between 10 to, and the biggest female is like 20 millimeter. Yeah. And they are really cryptic, and they live in places that it's so hard to find them, and they mostly live under the stones and rocks, especially the big ones, that you can, it's so hard to move these rocks and look under these rocks to see them. And they are super fast, so whenever you just turn the rock, they just go to other side, and it's super hard to catch them or see them. So these Holoptilon, it belong to family Gonipetidae. And so prior to my study, there was 3 known species. One, Holoptilon posilolum, which was first discovered by Bayer in 1964 from Jerusalem. He just found like some specimen, and he described it as Holoptilon posilolum. And there was not much finding or recording of these species until 2007, when Ahmad Katbebar and Abu Jonan, they find also some specimen in Jordan, and they identified them as these species. And but from the type locality, there was no record till 2018. Then they found this on Jerusalem again. And at the same time, in Iran, in north central of Iran, also Kolnigari finds some specimens, and he described another species called Holoptilon brevipugilis. And recently in Turkey, they also find like some specimen, and they said, okay, this is also a new species, and they just described it as Holoptilon yagmor. Then I was like in the field, and I tried to collect like a lot of material from so many places of Iran. And you can see in these photos, these are the habitat that you can find the Holoptilon. And I could gather like so many different specimens, but there was like a lot of problems I faced when I wanted to identify my specimen according to those previous studies and the description of these 3 species. What did I face? So there are like these characters that I put like these photos in these slides were like the most important characters according to the previous studies, that these can be used as a diagnostic character for these the species in Holoptilon. But what did I face? It was like when I looked into the thorax or like mesonotum and metanotum shape or width or length, all I see was like the variability. When I look at the femur, the spot or like the coloration, and all I see was like even in the specimens that I found like in one place at the same time, they had completely different. So do I have like so many different species, or are they one species? What do I have? And also like in tibial spines, because they were like describing the species according to if it has like 12 or 11 or 13 spines, so they belong to different species. But when I was like looking into that, according to this graph that you can see for like different specimens, it can even be variable within one species, like the femur of like the right and the left can have like different spines. So this is not a reliable character, morphological character. So I faced so many variability within the morphology. Then I was saying, okay, what should I do? I tried to look into more details and look into all possibilities for the morphology to find like something that is constant and can be reliable to say, okay, this character can be used to distinguish this species from the other species. Therefore, I tried to extract the male genitalia. Male genitalia is one of the characters that is used in Mantodea to identify different species. And I tried to look into the male genitalia, and I could find some characters that more or less can help to distinguish different species. And also, I find that in a specimen from one species, this character, this 5 spine is constant in all specimens, and it's reliable, and I can distinguish this species from all other Holoptilon species which had like 4 spines. But this was not enough for me. So we find like this in morphology, and then we said, okay, let's look into other data. Then we did the genetic study. I amplified like 4 genes, 2 nuclear and 2 mitochondrial genes, and this is the result. You know, when we try to make the phylogenetic trees, both maximum likelihood and Bayesian trees, they all had the same and identical topology. And when we look into the morphospecies, they were all founding, like we found all of them in monophyletic clades. So we could say, okay, we have like these morphospecies, this morphospecies. So we had some data on morphology, we had some genetic, and when I was like in the field, I could gather so many ecological data, and then I also tried to collect some and breed them and look into their life cycle. Therefore, I could implement all these data together. In other words, like I use integrated taxonomy to realize what exactly I have in Holoptilon. And it leads to discovering and description of 4 new species for this genus: Holoptilon abdullahi, Holoptilon iranicum, Holoptilon khuzestani, and Holoptilon taduaniensis. So what we wanted to do. So when we look at these, and when I was like in the field, I could see that, you know, these data and material that I could get, it's from 10 years of searching for these. And whenever I was in the field, it was so hard to find them. They are like, it's so rare to find them. And in each year, maybe I could find like 2 specimens or 3 specimens. So I realized there should be something, I should do something for their conservation and look into their conservation status. Therefore, I said, okay, when I am like describing new species and I am writing this paper, it's better to have like a preliminary assessment for these species, that these newly described species, so I can make like more attention. And when I just publish this paper and it's like in other social media, I can get more attention to these and talk about their status, and maybe I can make like some conservational impact for these things. So according to all the data that I could gather, I tried to do the preliminary assessment for them. And for like these species, Holoptilon poecilulum, we assessed it as endangered, and according to, as you can see, like this species is very localized. You can find them only like in Jerusalem and Jordan. And finding them was so hard, like the record is so scattered, it's like, it's not continuous, and yeah, and they can find like in very limited locations. So we assess them as endangered species. The other species was Holoptilon abdullahi. As you can see, it's only found in a very localized area, like in southern part of Zagros mountain, this green dot, and yeah, they are also assessed as endangered species. The other one was Holoptilon brevipugilis. We find them like in different localities, and but at the time that I was like writing my paper, we didn't have like any data in these question mark places, and we didn't know if the distribution is continuous or not. And but now I got some material from this area, this gap, and now I know that the distribution is continuous, and we assess these species as least concern. Another species was Holoptilon iranicum. This species also we assessed it as endangered due to these criteria that we had, and Holoptilon khuzestani as well. As you can see, it's really very localized, and all of these species, they are because you know they are like small mantids and they are wingless, and they just when they are like completely in one limited area they can be found, and when you are like in the field, it's not like you can find them like in so many different places. It's the population is so patchy, and when you know you find them like in here, it's only in here, and you cannot find them like in other places, and they are so specialized. And this species, Holoptilon tadouaniensis, unfortunately we couldn't get more data on it, and we just found it in one single locality in Tadouan. And because there are no further data on it, we just assess it as data deficient. And when we can, as you can see in this photo, this is the satellite map for like 2 species of Holoptilon. One of them is Holoptilon posilolum, and the other one is Holoptilon brevipugilis, from like different places that we find them. And you can see how much the urbanization and the degradation of the habitat can really threat these species. And yeah, when I was like in field, I could see, like as you can see, in so many habitats that these species live, especially like those small patches that I know that these species live. I saw that the people try to move all the stones to somewhere else, and they put like fire to get like some agricultural like field for them and plant something. And this really was like on exactly the place that I used to find like Holoptilon and Holoptilon abdullahi, and then I saw that it was completely destroyed. And after like now I have like some people going there, and I asked them to try to find like and monitor their population, but it's been like 1 and 2 years that they couldn't even find like one specimen. And I am so worried about these species. And also when I look into the papers about the climate change and the changing in precipitation in Iran, because you know Iran is somehow like it's an arid place, and these days, according to this paper, like that Sadernia et al. Like in 2023, they just published this paper. They found out that there was like a huge decrease and significantly decrease in precipitation in in frequency of the light, moderate, and heavy precipitation in Iran, and it can really affect the Holoptilon as well, because these species are all the habitat that you can find them. They are also reliable on some permanent water source, and these little precipitation and these decrease in precipitation can also threat them, and also these habitat changes that by land use that humans are doing. It's also another threat to these species. And I believe that with like doing like preliminary threat assessment and put them in the papers when we are describing the new species can help for conservation, and also like we can when we have like these preliminary assessments, we can say, okay, we need to prioritize like the conservation and prioritize studies on these species from these places, and it can help. And I believe that also establishing long-term monitoring can also help, and also monitoring their genetic diversity and see if it changes through time or not. And so as you can see, this was like about Holoptilon, but I saw also like some poster in this Congress that they were like doing some discovering some locusts in Croatia, and it was also a wingless and somehow a small grasshopper, and this also found to be endangered. So I believe that when we are like trying to find like different like species, it's good to first use like integrated taxonomy to understand what do we have, because in a Holoptilon case, which can be a model for other insects as well, when we look at the genus, it's like really hard from taxonomical view. We cannot know how many species we have, and if we cannot say, okay, because some people may say that this is one species, because when there is no much of a study, no data, we could say, okay, there is one species, and if this is only one species in all these places, we cannot really assess if it's endangered or not. But when we try to describe like and understand what do we have in like this cryptic genus, and we find out that these are different species and they are like endangered, we could use like some conservational program or like management to conserve them. I mean, for like insects and like something like Holoptilon or like mantids, maybe it's a bit hard to ask for like do the you know, prioritize like and put the national park for like these to conserve them, but still we can do some awareness and do like such these things to somehow make like some effect to conserve them, and not let these species before like getting you know, to know them, they get extinct. And this would be a really sad story to have them like get extinct before they get discovered or like being like introduced to the world. And with that, I would like to thank you for your attention.
Thank you very much for a very nice lecture. Questions?
Since Holoptilon is hiding under stones, do you think it might have a nocturnal behavior? In Spain, you have some, for instance, Perlamentis, the genus, which was thought to be extremely rare.
Yeah.
In recent years, there are a fair amount of new data, and they're also attracted
to the light. Yeah. I tried to, you know, it's been like 10 years of searching, and I tried to look under all the stones, and I tried to like look everywhere that I could. I also did the light trapping, but these species, you know, Perlamentis has they are fully winged mantids, and they are attracted to the light. Holoptilon, I also saw them that they are, you know, active during the night. And somehow where I had like some light trapping, and when not just going to the light, you know, when I was searching around, I could also find them. But the rarity of them, it's not just because they are like cryptic and living under the stone, or like so hard to find them. Yeah.
Axel?
Thank you for the nice presentation and the enthusiasm you have for it. It's really great. I think one important thing is that we as scientists, we should be aware that scientific papers are mainly read by scientists. So
Exactly.
Which is somehow showing this, that they are more or less responsible for these species. It can help a lot.
Yeah.
But then you need somebody who cares for them on the ground. Yeah, because,
Yeah, this is also, I was like, I am trying to do like, in also like, I have an Instagram account and I try to, now I want to like put now that the paper is out and people are like getting to know it. So I want to post like, and because I see like there were even some photographer in like these areas and even from some area that I wasn't aware of that Holoptilon exists there, they were like saying runner mantid, very fast runner mantid, and they took a photo and put them on the Instagram, and this way I could find, okay, this is the new locality for Holoptilon. I believe that in social media we can also post like about these amazing and saying that they are endangered and put like some photos and talk about it and saying that these are like legacy for like these region, and yeah, we can make like some awareness and make these people, because there are like a lot of people that they are not scientists, but they love nature, being in nature, and photograph the wildlife and nature. We can draw attention to them and make them to care about this. Yeah.
Thank you very much. Just one more observation. I think Holoptilon tadovaniensis should be tadovaniense because it's neuter gender.
Ah, okay.
And it also checks for brevipugilis. Maybe it should also be brevipugilis.
Yeah, brevipugilis is not my species, but yeah.
Yeah, so you can correct it in the next
Okay.
paper. Thank you very much.
Yeah, thank you.
Now we go a little bit northwest to Greece. Not to Greece, sorry. A little bit west to Spain. And next speaker is Joaquin Ortega, and he will speak about Podismini grasshopper.
Is the pointer? Where is the pointer?
The pointer, I'm not sure. Filippo, where is the pointer?
Pointer? Sorry. Here. I see. Okay. And now it's working. Thank you. Thanks, Joseph, and thanks to the organizers for this conference. It's my first time here, and it's been great to meet so many people in person finally. So I'm going to present here some preliminary results on our ongoing project on integrative taxonomic, demographic, and extinction risk assessment of Podismini grasshoppers from Mediterranean sky island archipelagos that include many Orthoptera that are extremely vulnerable to climate warming. So basically, this project with the acronym ALPIMATE is based on the study of 3 in situ radiations of Podismini grasshoppers, including Podisma from the Iberian Peninsula with 3 species and 2 subspecies, Italopodisma from the Central Apennines with a total of 9 putative species and 5 subspecies, and Oropodisma in the Balkan Peninsula with a total of 10 species. Here I'm going to talk basically about Oropodisma, a little bit on Podisma, and our preliminary results on Italopodisma have been presented by Francesco Forte in a poster that probably many of you have already seen. So I'm going to focus mostly on Oropodisma here. So basically, all the taxa within the 3 radiations share some common attributes. They are, as I mentioned before, they are alpine species. They are cold-adapted. They have very low dispersal capacity. They are either wingless or they have very short wings, so they cannot fly. They are, in most cases, narrow endemic taxa. They live in a single mountain range, and in some cases, we only know a single locality. And they have a controversial taxonomy. Most of the species within the 3 radiations were described on the basis of subtle differences in internal male genitalia. So this taxonomic hypothesis have never been tested using genetic or genomic data. So there is a controversy about how many species are included actually within each radiation. As I mentioned before, because they are alpine species, today they live on mountain tops, usually about 1,800 meters of elevation. And because they have very low dispersal capacity, most populations are highly fragmented today. Most likely that in glacial periods in the Pleistocene, that in the coldest stages of the Pleistocene, most of these populations went down in elevation. So they probably come into contact in mountain foothills and valleys where they probably exchange inflow. So most likely they sustained much larger effective population sizes in the past than today. And as a consequence of climate warming, we expect that most of these populations experience upslope distributional shifts, going up in elevation and potentially with some local extinctions in mountain range at the lowest elevations. Because of this, most of the taxa within the 3 radiations are included in the red list of the UCNN with different categories, including near threatened, vulnerable, endangered, and even critically endangered. So basically the objectives of this project, Alpinet, is to do first a taxonomic assessment, like integrating genomic data and morphometric data, phenotypic information, to evaluate the taxonomic status and evolutionary distinctiveness of the different taxa within each species radiation. We also want to do an evolutionary biogeography assessment trying to infer the processes that have contributed to the high levels of local microendemism in these 3 radiations. We also want to do a conservation status assessment, like documenting recent distributional shifts, comparing historical records and contemporary transits we have been doing that in past years. And finally, we want to do a demographic and extinction risk assessment, like determining the factors that have shaped the demographic history of each species, and use these inferred demographic parameters to forecast the fate of the different species and populations under different scenarios of climate warming. So basically what we are doing is to sample pretty much every known population of every species within the 3 radiations. Here I show you the distribution of the genus Podisma in the Iberian Peninsula and the populations that we have been sampling. We usually sample around 8 individuals per population. We extract the DNA and prepare RADSEC libraries. I'm not going through the details, but and finally we treat the data bioinformatically using this program, IPyDAT. So at the end, we get between 5,000 and 10,000 loci. We are also obtaining morphometric data. Here I show you the furculi of 2 taxa of Oropodisma. And basically what we are doing is to quantify these differences in morphology using geometric morphometric approach. And finally, we are building environmental niche models to predict or to understand, to infer distributional shifts of the different species and genera from the last glacial maximum to present. So basically the idea of this project is combining all these sources of information like distributional information, genomics, morphometrics, and environmental niche modeling to answer the different questions and to address the different objectives. So I'm going to present here some very preliminary results regarding taxonomic assessment. One of the things we could finish is to build a phylogeny of pretty much every known population of Oropodisma from the Balkan Peninsula. We sample, I think, 34 populations, and we built a phylogenetic tree using SVD quartets and RAXML, and we dated the tree using BPP, considering the mutation rates for Drosophila melanogaster. So as you can see here, and the topology of the tree is very similar to the one presented by Nefeli Kotitsa based on mitochondrial DNA. What we found is a clade here that is the one in the Peloponnese, another clade in the north, including most of the species out of the Peloponnese. And as you can see here, well, there are different populations that belong to the same species, like Oropodisma macedonica, that is the most widespread species, Oropodisma carabica, etc. And there are some populations here indicated in red as OSP that are populations with an unknown taxonomic status on the base of the papers by Fer Willemsen and Luke Willemsen. So basically, well, as you can see here, according with our dating, most lineages started to diverge like 2.2 million years ago. And on the base of this ultrametric tree, what we tried was to do a species delimitation approach using this program, Delineate. And so we can assign some species, some populations to some species. For instance, these two populations on the top here, OSP, they were assigned to Oropodisma macedonica. The same thing for these populations here that were assigned to Oropodisma carabica. And we found one lineage here, highly divergent, that is probably a cryptid species, a new species. So there is still some diversity to be discovered within the genus Oropodisma. Oops. It's not working. I think it got stuck.
It's stuck.
Oops.
He will do it.
Okay.
He will show.
Oh, perfect. So it's someone. Okay. Thank you. Another thing we have been working is on distributional shifts of the different populations. And here I show you a map with the distribution of the different species of Podisma in the Iberian Peninsula, include Podisma cantabrica. It's an arrow endemic in the northwest, in the Cantabrian Mountains. Podisma pedestri, that is more widely distributed in the Pyrenees. And Podisma carpetana with two taxa, Podisma carpetana ignati in the north and Podisma carpetana in the central system, very close to Madrid city. And here I'm going to focus mostly on our results on Podisma carpetana. Basically, what we did was to compare historical records of species distribution of Podisma carpetana in Sierra de Guadarrama based on museum specimens and literature records, and we compare with contemporary transits that we have been doing past years to see if the species has experienced elevational shifts and range contractions, probably as a consequence of climate warming. I forgot to mention that these populations are the southernmost populations of any Podisma in the Western Palearctic, so they are expected to be particularly vulnerable to climate warming. So basically what we found is that the species has experienced an important upslope distributional shift. Here I show you in deep blue the minimum, the areas occupied by the species on the base of the minimum elevation at which this taxon was recorded at the end of the 19th century and the beginning of the past century. In light blue, the same thing, but between the 50s and the 70s of the past century, and in red, the minimum elevation areas at which the species has been recorded today, basically today. So you can see that the species has experienced, according with historical records, an upslope distributional shift of almost 900 meters of elevation. And actually, according with our transits, the species only persists today in a small habitat patch, less than 0.1 kilometer square. Here I show you circle size is proportional to the number of specimens we found in our transit. So they only persist in a very, very small habitat patch. So these populations are at the verge of extinction, probably. And this is in line with our findings for genetic diversity, again, based on RADSEC data. Here I showed you the distribution of Podisma carpetana across the Iberian Peninsula. And red color means high levels of genetic diversity, blue color means low levels of genetic diversity. The populations in the south, in Sierra de Guadarrama, have the lowest levels of genetic diversity across the entire distribution of this taxon. And in fact, what we found is a positive correlation between genetic diversity and latitude. That is exactly the opposite that we found in most organisms, in which we found usually that the southernmost populations have higher levels of genetic diversity. Here we found exactly the opposite. And pretty much the same thing we found for the southernmost distributed populations of Oropodisma in the Peloponnese region. That is the southernmost limit of the genus distribution. Here I show you the distribution of the species in the 70s, again based on the minimum elevation at which the species was recorded by Fer Willemsen in the 70s. And in red, the minimum elevation range at which we recorded the species in 2020. You can see that this species, Oropodisma ribantosi, has experienced an upslope distributional shift of almost 300 meters of elevation. And pretty much the same thing happened for other species like Oropodisma quilini, exactly the same for Oropodisma kelmosi in Mount Mainalon and Mount Parnon, and the same for Oropodisma taigetosi. So we have, I think, we have to work on the reassessment of the conservation status of these species because, for instance, Oropodisma taigetosi, I think, now is classified as least concern, if I remember well, but the populations there are almost extinct. I mean, in 2 days of intensive sampling, we could only find a very, very few individuals, like 4 nymphs or something like that. So many of these populations are, again, at the verge of extinction. Another thing we could do is to build an environmental niche model for the entire genus Oropodisma. This is a distribution model for the whole genus, not for a specific species. So basically the y-axis is the number of cells occupied by the species or the area. Y-axis is the area occupied by the species, and the x-axis is time. So 0 is present, and at the end it's the last glacial maximum, like 22,000 years ago. So you can see how the areas occupied by the species have decreased progressively from the last glacial maximum to present. These are reconstructions based on 100 years intervals. So in this map, you can see the present distribution for the whole genus Oropodisma. Obviously, all the populations today are highly fragmented, as expected for an alpine species. This was the predicted distribution around the Holocene, 12,000 years ago, more or less. Populations were more connected than today, and populations that in the last glacial maximum, as expected, were much more connected than today, probably because they went down in elevation and all the different populations were much more connected. Because of this high fragmentation of populations today, they also show a high genetic fragmentation. Here I show you the results for Oropodisma macedonica. You can see that pretty much every color is a different genetic cluster, and pretty much every population is assigned to a different cluster with some admixture, but still a lot of genetic structure. And pretty much the same thing, pretty much the same thing happened for Oropodisma kelmosi in the Peloponnese. In this case, with no signatures of genetic admixture. So populations from Oropodisma kelmosi are even more fragmented than those from Oropodisma macedonica. And finally, we could do some demographic reconstructions. For here I show you the 2 examples for Oropodisma kelmosi and Oropodisma lagrecai. The y-axis is effective population size, the x-axis is time, and the vertical dashed line is the last glacial maximum. So you can see here how the effective population sizes have decreased from the last glacial maximum to present, as again in line with the expectations from the environmental niche model and as expected for an alpine species. So basically, what we found, some preliminary conclusions, is that the taxonomic status of the different populations must be carefully re-evaluated. Many populations have experienced important upslope distributional shifts. Populations are genetically highly fragmented today, and effective population sizes have continuously declined from the last glacial maximum to present. So that is it. Thanks to my different collaborators, Ana Papadopoulou, Mercedes García París, José Miguel Aparicio, and Victor Noguerales. Funding coming from the Minister of Science and Innovation from Spain, and thanks to the different countries and environmental agencies that granted us the permits for sampling these grasshoppers. And that is it. Thank you very much for your attention.
Thank you very much for the amazing lecture. We have time for one question because lunch already started. Please.
Paula.
Sorry, Paula, I didn't see.
Oliver.
First of all, compliments for this wonderful presentation. And what I was thinking about these species that are affected by increasing of temperature, we know very little things about their biology, especially what they eat. Do you have some information about this? Because animals, they reproduce, but they eat. And so it's a very big lack in the knowledge of most of the species we are presenting here in this Congress. So what I suggest is to spend some efforts to investigate this aspect. That, in my opinion, is not absolutely of secondary relevance.
Yeah, I agree that it's important. I have a colleague from Spain, Paola Laiolo, that she did some metabarcoding to identify the plants that this Podisma carpetana, I think. And what she found is that they eat pretty much everything is around. I mean, they are not very specialist, apparently, according with her data. So, but she never published that, I guess, because she didn't find anything like very exciting or something like that. But
And another aspect is to cross the data with botanical data.
yeah. And Bruno.
Is there some genetic difference between Carpetana and Ignati?
We published already a paper about the phylogeny of the different Podisma in the Iberian Peninsula. And, I mean, Carpetana Ignati is paraphyletic, or in other words, Carpetana is within Carpetana Ignati, and other populations are more in the base of the phylogeny. So there is no phylogenetic support for the subspecies Carpetana carpetana. That is the point, I guess.
Thank you very much. We continue discussion on lunch. Is it okay?
Great. Thank you.